We're restoring a landfill site that contains construction rubble from a demolished building into a native meadow grassland. The site is just under 1 hectare in size. In 2014, the college took down Bowman Hall on campus in order to make room for the new Krager Center. The rubble from the residence hall was placed here, and the area was terraced flat. It was seeded with a mix of grasses and clover. Over the years, the area was used to store materials removed from campus. The site was sparsely vegetated with weeds and invasive species. The soil was compacted and rainwater pooled on the surface. In this video, I'm going to explain the steps we went through to go from rubble to meadow. Let's get started. In the fall of 2020, the environmental studies seniors proposed the idea of taking this degraded, underused area of campus, restore it with native vegetation, and to use the area as a living laboratory. Why restore the site with native vegetation? First, grassland species have roots that can penetrate up to 2 meters into the soil. The roots will work around the buried debris, improving stormwater infiltration and increase soil carbon storage. Second, communities of native plants fuel the food web, promoting populations of animals that include species that pollinate, eat pests, and have conservation concerns. Just months after the seniors proposed their idea, a group of faculty took on their idea. They developed a scope of the project and gained approval from the academic dean, to the president's council, to the board of trustees. Along the way, 2 generous members of the board invested in the Environment Center, giving us seed money to get underway. In the summer of 2021, a group of students and faculty researchers began analyzing the soil, and they conducted a biodiversity blitz, evaluating the presence and abundance of plants and animals. To do the meadow restoration right, we needed resources. In January 2022, we submitted a grant proposal to the Community Conservation Program at the Virginia Environmental Endowment. We heard on Earth Day that our grant was funded. Along the way, we dove into the literature to learn from others. We discovered the truism that every restoration project has a unique set of conditions and constraints. So there's no one-size-fits-all prescription. Nonetheless, there are 3 common challenges to restoration work such as ours. The first is dealing with the low quality of degraded soil and the high level of soil compaction. Therefore, our soil assessments and planned soil ameliorations focused on these attributes. Second is the continued challenge of controlling invasive species. Our vegetation assessment showed that the site was dominated by invasive species that are notoriously hard to remove. We chose an integrated approach to invasive plant management. Throughout the process, we used mechanical methods of cutting, mowing, hand weeding, and tilling, as well as, when needed, targeted herbicide. The third challenge is white-tailed deer herbivory will reduce plant diversity. Deer heavily use the adjacent woodlot for cover and come into the meadow site daily to feed. Therefore, the native plant species that establish and persist will be those that can tolerate deer herbivory. Using what we have learned in the literature and with our VEE grant in hand, we redoubled our efforts to learn as much as possible about our site. We did a more thorough soil assessment and more biodiversity work. Now we were ready to start the restoration. Our first step was cutting the woody vegetation, including black locust trees, and apply herbicide to the site. The next step in our plan was to improve soil quality and lessen compaction. To do this, we planned to loosen the top 10 to 15 centimeters of soil and mix in compost and topsoil. We discovered, because we wanted to disturb the soil in an area larger than an acre that was not zoned for agriculture, that we would need to obtain a stormwater management construction permit from the state. We hired a firm to help us, and we worked closely with our local municipality. In the late summer and fall of 2022, while we waited for our permit process to be completed, we pivoted our plan. We had enough topsoil and compost piled at the site to cover the south half of the area, so we hired a contractor to put up a silt fence and use a small bulldozer to spread the material without disturbing the existing soil. We then seeded 20+ species of native grasses, forbs, and nitrogen fixers. We also added a cover crop of cereal rye. Over the late fall of 2022 and winter 2023, while the permitting process continued, we moved enough leaves collected from campus to use as compost that would cover the north half of the site. Also during the winter, we held a campus-wide service event. Students, staff, faculty, administrators, and community members came out in force. Volunteers sorted and stacked pavers that had been stored at the site over the years. Now these pavers could be reused and kept out of the landfill. We also installed a fence that delineated the site and restricted access. Come spring 2023, the cover crop grew and dominated the biomass on the south side of the site. Nonetheless, still, invasive plants were abundant and rainwater pooled. Only a handful of plants that we seeded came up. In the summer of 2023, we continued with our biodiversity research that will allow us to assess if and how the meadow restoration work will impact animal communities. The cover crop died back. Invasive species thrived throughout the site, and a thick cover of invasive Johnson grass covered the mound of compost. Therefore, we mowed the entire site and selectively sprayed herbicide on the compost mound. With our permit in hand, we could now do the major soil work in the summer of 2023. We hired a contractor to loosen up the top 10 to 15 centimeters of soil, spread compost, and work it into the soil. The spreading and mixing of soils stimulated the seed bank. Invasive species grew thick and quick across the site. This allowed us to treat and remove the invasives before we planted. On the south end of the site, we invited volunteers to hand pull and cut the invasive plants 2 times over a month. On the north half of the site, the Johnson grass was just too thick for hand weeding. Here we hired a contractor to mow and use herbicide. We let the area lay for a month, then we did a final treatment of mowing and tilling. This knocked back the weeds one final time and loosened the soil, providing an uneven surface for the seeds to come in contact with. We had 24 species of native plant seeds. We divided them up into 8 equal amounts in Ziploc bags and combined species based on seed size. We used flags to divide our site into 8 evenly sized sections. This made it easy for students to spread the seeds. We also seeded cereal rye as a cover crop. We took one more set of soil samples and measured depth to resistance, a measurement of compaction. Our soil analysis showed that adding compost and other material increased the total carbon and nitrogen, and the soil pH was between 7 and 7.8. So we did not need to do any further soil amelioration at this time. Over the winter, there was very little change. In March, we installed data loggers that measure air temperature and soil moisture and temperature. These sensors, along with our weather station, will allow us to monitor how plant growth, temperature, and season alter stormwater infiltration. In April 2024, the site was greening up with enough vegetation that our permit was terminated. We could remove the silt fence. The students were able to pull the fence out of the ground, completely removing it, rather than cutting it and leaving the buried part on site. In May, the cover crop was growing well. The vegetation was heterogeneous. The amount of biomass, invasives, and native species across the site varied. To promote the native plant's ability to establish and thrive, we will bush hog the site several times this year to cut back the cover crop and invasives allowing more sun to hit the small native plants as they establish. The restoration work at this site will be ongoing. Our goal is for our meadow to be a place where native species thrive. Non-natives and invasives are here to stay and will always be part of the community. Another goal of our restoration was for this area to be a living laboratory and involve as many students from different career paths as possible. Through their hands-on experiences, they have learned about the how, what, and why of environmental restoration, and they will take that with them after Roanoke. Thanks for watching. Follow our progress on our YouTube channel.